A new paper came out last week on DNA methylation. DNA methylation falls under the broader category of epigenetics, and epigenetics falls under the broader category of genetic regulation. Now, epigenetics and DNA methylation have only been understood for a few decades now, but they are revealing an unbelievable level of complexity in the cell. You can think of DNA methylation as opening and closing certain sections of DNA for gene expression. At least that's the textbook explanation these days. But this new paper is revealing more roles, more functions of DNA methylation. In addition to this kind of tactical role of opening and closing sections of DNA, DNA methylation also influences the genome architecture. All of this is a monster problem for the theory of evolution. Let's have a look. Here is that paper, but first a little bit of review. Here's the DNA structure, or a rendering of the DNA structure. Of course, everyone knows it's a double helix or a double coil. In other words, it's kind of like a string where you have two threads wound around each other, each one forming a coil. Now, that's just a fancy artist's rendering. Each thread is actually consists of nucleotides, and here's an illustration of 4 of the nucleotides bonded together. There's thousands of these nucleotides, of course, forming each thread in the DNA double coil. And the C stands for cysteine, G guanine, A adenine, and T thymine. So 4 different types of nucleotides are used throughout, creating each DNA strand chemically bonded together like this. So you have this double coil. Now, that coil itself is wrapped around some proteins. These proteins are called histones. They're in the middle of this chart. The DNA double helix is wrapped around this group or cluster of histone proteins. This structure where the DNA is wound around the histones is referred to as a nucleosome. And you have one after another nucleosome as the DNA wraps around one and then another and another. And you have kind of like beads on a string as the DNA is connecting all these nucleosomes. Now you'll notice that we had coiling and then we had coiling again, right? The DNA double helix was coiled, and now it's coiled around these histone proteins. So we have supercoiling going on. It's going to happen again, and this chart illustrates this hierarchy. So on the far left, you have the basic DNA double coil. Then next, that's wrapped around the histones to form the nucleosome. So that's our second level of coiling. And now you have your beads on a string. Now those beads on the string are going to be coiled around themselves. So now this is the third time we have coiling, and so on and so forth. So you have multiple coilings going on. It's supercoiling. And you finally end up with this packed DNA structure. It's an incredible packing scheme is used to store the DNA. Of course, you have to get at the DNA somehow, so let's talk about that. This is where the methylation comes in. So we have the nucleotides here forming the DNA thread. Now consider the adenine here, the third one down. Let's zoom in on that. And here's just that side part of the adenine. You have a couple of rings here. Notice this nitrogen here at the top. Let's zoom in on that. That has 2 hydrogens attached to it, and the nitrogen is attached to a carbon down here in the ring. So let's illustrate that here. So here's the carbon, the nitrogen, the 2 hydrogens. Now consider a simple methane molecule. Methane is one of the most simple organic molecules, a single carbon and 4 hydrogens, CH4. Let's put these 2 together here. So here's the top of that adenine nucleotide, and here's our methane molecule. These 2 hydrogens, let's get rid of those. And when we get rid of them, we can bring the methane over and bond it to the nitrogen. And there you go. So we've attached a methane to the nitrogen here. In the process, we had to get rid of one of its hydrogens. So we call this a methyl group. So it is now a methyl group is now attached, and that's called methylation. So we have methylated the adenine, and here's what it looks like in this diagram. So we were looking at the NH2 there at the top. It now is just an NH. You drop off one of the hydrogens, and instead replacing it with this CH3, this methyl group. that's methylation. So when you see a CH3 show up, you know that's methylation. Now, there's several complications to this methylation process. First of all, the cytosine nucleotide also can be methylated. In this case, the cytosine can be methylated in two different ways. So here on the left, you have your basic cytosine, and then we have two different methylated versions. In the first one, you see the CH3 there in the 10 o'clock position, and then secondly, it's attached to the nitrogen again, like in the adenine case. Two different ways to do the cytosine. Now next, those histone proteins can also be methylated. You see these tails sticking out? So you've got the histone proteins in the core here, but they have tails sticking out. Those tails can also be methylated. Thirdly, there's other molecules in the mix here. It's not just methyl groups. Several other small organic molecules can be used as well. So for example, acetyl groups can be attached. Next, these molecules can be added or removed, and this is done by proteins. So there you have proteins going around attaching methyl groups or acetyl groups or other small molecules to the DNA or to the histones, and they can be added or removed. These molecules can activate or repress. So the methyl groups can activate that section of DNA or repress that DNA. Now, when I say activate or repress, I'm referring to kind of like opening it up or closing it down for use. So the genes in that section of the DNA are more likely to be expressed. Copies of them will be made, and that information will be used. That copy will be used, for example, to make proteins. Now, I know what you're thinking. How can we tell whether it's activated or repressed? Well, it all depends. It depends on what molecule is used and where it's used. Are we talking about methylation? How much methylation? Where are the methyl groups added? And so forth and so on. So it all depends on the situation. It's very complicated. Also, it depends on additional molecules. You can methylate a methyl group. So you can add a methyl group, and then that methyl group can be methylated. So there's a hierarchy to these molecules. So, okay, with that background, back to our report here, and let's look at the title because it kind of says it all here. DNA methylation is required to maintain both DNA replication timing precision and 3D genome organization integrity. Okay, what does that mean here? DNA replication. All cells need to make a copy of their genome. That's called DNA replication. When that happens and how that happens is very precise. It needs to happen at a certain time. And the DNA methylation is required to maintain that precision. It also maintains the genome organization integrity itself. In other words, it's doing a lot more than what I said a minute ago. Remember, I just said the methylation opens and closes sections of genes. It allows the genes in those sections to be expressed. Okay, good. That's kind of a basic function. This paper is shedding light on much more. Methylation is doing much more, and that's interesting. Skipping down, let's read from the discussion section. DNA methylation facilitates the maintenance of replication timing and higher order genome architecture. We show that genome-wide loss of DNA methylation is associated with a remarkable reduction in replication timing precision and corresponding loss of 3D genome compartmentalization. Our findings suggest that DNA methylation is not only associated with transcriptional regulation, but also plays a critical role in the maintenance of higher order genome architecture genome-wide. Wow, so this methylation is really doing a lot. Now here is an article about this paper, and skipping down, a researcher not involved with the research made this comment that, gee, there are regions of the genome that are hypersensitive to these fluctuations, and she's referring to these methylation fluctuations. So this is very precise. Now, so we can add to our list of methylation complications a couple of more bullets here. Methylation is required for timely replication, timely DNA replication, and methylation is required for higher order genome organization. Things just got a lot more complicated. And this is going to be a problem, a big problem for evolutionary theory. Let's work through this sequence here. Under evolutionary theory, if you're an evolutionist, you have to believe something like this sequence is going to happen. DNA somehow evolved. The genetic information in the DNA, the genes, somehow evolved. The cellular machinery around the DNA to use that information to express the genes, that is, make copies of the genes, and then make proteins using that information, somehow that evolved. Some proteins perform the methylation tasks to activate the genes. So you've created these proteins now. You're using the genes, you're making proteins. Some of those proteins are going to do that work, do that methylation work. There was that removing and adding the methyl groups to activate or deactivate the genes. So now the genes can be expressed to make proteins, and now the genome can operate properly because of this methylation. But wait a minute. Something's wrong here. I said up here in item number 3 that you were going to make proteins. So you're making proteins that can now do methylation tasks so that you can now make proteins. So let me go to another slide to try and explain this a little better here. In other words, special proteins are required for methylation, and methylation is required to make proteins. You have a chicken or the egg problem. And I have a little asterisk here because methylation is not only required to make proteins, but now this new paper is telling us a lot more. Methylation is also required to make the cell function properly. We have a huge problem here under evolutionary theory. Too many things, way too much complexity is required all at once to get this thing up and running. There's no hint of a gradual pathway, one step at a time, one mutation at a time to get this thing going. You need methylation, and you need it badly. It's just another example of how biology contradicts evolution. The science contradicts the theory.